Dear student, the topic of this video is ovarian hormone. Once luteolysis begins, the estrogen and progesterone levels fall. This is a picture we have seen several times while discussing the endometrial cycle and the ovarian cycle. So once luteolysis begins, that is, luteolysis will start from the 24th day of a cycle. So the estrogen and progesterone level, they will gradually fall, and the secretion of LH and FSH will increase. Okay. So after this phase, when a new cycle will start, so the LH and FSH levels gradually it is high due to lack of estrogen and progesterone, and a new crop of follicles develop. We know that from the primordial follicle, they will become the Graafian follicle. Near mid-cycle, estrogen secretion from the follicle, it rises. So this follicle, growth of the follicle will be facilitated by this FSH. Now from this follicle, there will be secretion of estrogen, and estrogen level, it is gradually increasing. In the mid part of the cycle, it will reach its peak. Now this rise augments the responsiveness of the pituitary to GnRH and triggers a burst of LH secretion. You can see that this phase it corresponds with the burst of LH secretion, that is the LH surge. This results the ovulation. This results the process of ovulation. And after that, after ovulation, there will be formation of corpus luteum. Now during this period, estrogen secretion, it drops. But progesterone and estrogen levels then rise together. Both the progesterone level and the estrogen level, they rise together. Okay, because it will be secreted from the corpus luteum along with the level of the inhibin B. The elevated levels of this estrogen and progesterone, they will inhibit the LH and FSH. So you can see in the luteal phase, the value of LH and FSH, it is very less. How long it will remain less? Until and unless the corpus luteum will start undergo regression, and there will be drop or decline in the level of estrogen and progesterone. Again a new cycle will start where there will be again increase in LH and FSH level, and the cycle will be repeated. So from this we have encountered with many hormones. Okay, we have encountered with the LH and FSH, we have encountered with the estrogen and progesterone. Okay, so this LH and FSH, as we all know, they are secreted from the anterior pituitary, and from the ovary, the ovarian hormone are the estrogen, mostly the estrogen and progesterone. So today the topic that we are going to discuss is the ovarian hormone. So the name of the hormones are the estrogen, progesterone, relaxin. So when we will start discussing any hormone, so there is a common formula for describing a hormone. We have to know about the chemistry or the structure of the hormone, how it is synthesized. Second, how it is secreted, and after secretion, how the metabolism occurs, and when it reaches the target organ, what are the gross functions of that hormone, and the mechanism of action of that hormone. So grossly the 6 heading: their structure or chemistry, biosynthesis, metabolism and secretion, functions or actions, and the mechanism of action. So now we will try to describe the function of estrogen under all these 6 headings. The naturally occurring estrogens are 17 beta estradiol. It is also known as E2. It may be the estrone or E1 or the estriol, that is E3. All of them, they are C18 steroids. They are secreted from the granulosa cells of the ovarian follicle, corpus luteum and the placenta. And their biosynthesis actually, they depends upon the enzyme aromatase. Okay, so their biosynthesis, it will depend upon the enzyme aromatase, that is CYP19, which converts the testosterone, to estradiol. It converts the testosterone to estradiol. So aromatase will take part here, or it will convert the androstenedione to estrone. So here again the aromatase enzyme. So androstenedione will be converted to estrone. Okay, and this later reaction will take place, in addition to all these areas, it will also take place in the fat, liver, muscles and brain. So this is a very important concept, that is a two-cell theory of ovarian steroidogenesis. So in the theca interna cells, they have the LH receptor. Theca interna cell, they have many LH receptors. LH acts via CAMP to increase the conversion of cholesterol to androstenedione. Okay, so this intracellular cascades of reaction will convert the cholesterol to the androstenedione. But this androstenedione cannot be converted to the estrogen in the theca cell, because theca cell does not have the required enzyme aromatase or CYP19. Now, the theca interna cell, they supply the androstenedione to the granulosa cell. At the granulosa cell, they have many FSH receptors. So when the FSH will bind with them, so there will be increase in the intracellular CAMP, which will enhance the activity of this CYP19 or the aromatase enzyme. So androstenedione will be converted to estrone. Okay, and ultimately the estradiol, it will be released into the blood from the granulosa cell. It is seen that matured granulosa cell, they not only have the FSH receptor, they also acquire the LH receptor, and when this LH receptor will bind with the LH, it will enhance the estradiol synthesis. So this is another picture. So C27, it will form the androgen C19. Now this C19 will go to the granulosa cell, where they will form the C18 steroid. That is, after secretion into the circulation, the total estrogen, only 2% of it will remain in the free form. The remainder of it will remain bound with the bound form, that is around 98% will remain in the bound form. Out of that, 60% is bound with the albumin and 30% bound with the steroid binding globulins. Now, the concentration of estradiol in the plasma during the menstrual cycle, it is shown in this figure. We have also seen previously, the previous figure also. So here we can see the concentration of the estrogen. There are two peaks of the concentration of estrogen. You can see two peaks. One here, a peak here. Another peak is, this is the estrogen. Another peak is here at the luteal phase. Another is just before ovulation. Now, the estradiol secretion rate, if you see the 24-hour secretion of production rate of estradiol and the estrone, you will find that in the Early follicular phase. In the early follicular phase, the level of estradiol it is 36. The level of estradiol it is 36. And in the pre-ovulatory phase, it is raised to around 380 microgram. And mid-luteal phase, the value is 250 microgram. That is 250 microgram per day. It is here we are calculating the amount in 24 hours. Okay. So we can see there is increase in the level of estrogen. In the just pre-ovulatory phase, it will reach the value of 380, and again in the mid-luteal phase, the value is raised to 250. So we have covered the structure and of the estrogen, how it is synthesized, and how when it is secreted into the blood, what will be the level at different menstrual phase. Now it's a time to know about the metabolism of it, the function of it, and the mechanism of action of it. So in the liver, the estradiol, estrone, and estriol are converted to the glucuronide and sulfate conjugate. So in the liver, actually, in the liver, this estrogen, that is estradiol, estrone, and estriol, they will be converted to the glucuronide, that is the conjugated estrogen. You can see here, these are the conjugated estrogen. Now, all these compounds, along with other metabolites, they will be excreted in the urine. All these compartment, majority it will enter into the circulation, it will pass through the glomerular filtration, and there will be urinary excretion of this conjugated estrogen. But appreciable amounts are secreted in the bile and reabsorbed by the enterohepatic circulation. So appreciable amount it will be released in the bile, and through the enterohepatic circulation it will again be recirculated. This is the mechanism of metabolism of estrogen. So we can understand any kind of liver disease, that means the metabolism of estrogen will be less. That's why in liver disease, that is in alcoholic cirrhosis or in diseases like this, the male person may generate gynecomastia due to increased level of estrogen. It is not due to the increased production of estrogen. Estrogen metabolism will be less. So in that case, there will be gynecomastia or features of high estrogen level. Now the gross functions of estrogen, we have discussed it so many times while discussing the reproductive physiology. So in the heart, it protects from cholesterol. In the liver, it reduces the cholesterol in the blood, and in the vagina, it makes it moist and protects it from infection. Bones, it strengthens the bone, increase the bone density. Skin, they makes them young, and we can relate with the symptoms of postmenopausal symptoms where the estrogen level is less. So the postmenopausal women will have the weak bones, skin will not look like young like this. So in the breast, they grows and shapes the breast, prepare breast for feeding. In the brain, it helps the adjustment of body temperature, increases the memory, and adjusts the libido. And especially and more especially the function of estrogen in the uterus and ovary. It helps in the ovarian follicular growth. It increases the motility of the uterine tube. In the uterus, it does lots of things. It increases the blood volume, it increases the muscle mass, it increases the contractile protein activity, so muscles become excitable. So estrogen primed uterus, it is very much sensitive to oxytocin. And it is seen that female type of secondary sexual character, it will be produced by estrogen. Now one point is important here, but the prolonged so we know that estrogen is a kind of stimulatory effect, it has stimulatory effect in the uterus. So prolonged exposure to estrogen, for example, if A woman is taking an oral contraceptive pill containing only estrogen, no progesterone. So prolonged exposure to estrogen, or postmenopausally someone is taking the hormonal replacement therapy, that may cause the uterine cancer or endometrial cancer. So that is a risk factor of estrogen. Now come to the mechanism of action of estrogen. So estrogen, like all steroid hormones, then the receptor is a cytoplasm, the nuclear receptors. So they have the two principal types of nuclear receptor. One is the ER alpha, estrogen receptor alpha, another is the estrogen receptor beta. So estrogen receptor alpha, it is encoded by the gene on the chromosome number 6, and whereas ER beta, it is encoded by a gene on the chromosome number 14. So both these receptors, ER alpha and ER beta, they belong to the nuclear receptor superfamily. After binding with the estrogen, they form a homodimer. So when the estrogen binds with them, they form homodimer and binds with the DNA. Okay. So there will be transcription, there will be formation of mRNA, and mRNA from mRNA, so there will be translation of proteins. So ER alpha and beta, sometimes they form the heterodimer like this. So ER alpha, ER beta, this heterodimer they may form, or they may form the homodimer. Now most of the effects of this estrogen, it is genomic effect, that is, it is acting through the nuclear receptor. Okay, and we know the mechanism how it is happening. But so this is the location of ER alpha: uterus, kidney, liver and heart. ER beta, the uterus, kidney, liver. Here there will be ER beta and ER alpha. And it is again we can see that when it is acting over here, there will be release of mRNA. mRNA will cause a translation of proteins, and all these functions of estrogen we know in the uterus, in the cervix, in the brain, in the breast. So many actions will take place. Okay. Now, and all these actions are the genomic actions. Okay, they're working through the nuclear receptor. But there are some actions which are very rapid, that it is difficult to believe that they are mediated via production of mRNA, because the production of mRNA, then translation of protein, it will take some time. But these actions are very fast. For example, the neuronal discharge in brain, for example, the feedback on the gonadotropin hormones. Okay, we know that when the estrogen is released, it will give a negative feedback. So this negative feedback effect, it is very fast. So scientists say that maybe this reaction is not mediated by the nuclear receptor. Instead, there are possibility of cell membrane receptor. There are possibility of cell membrane receptor, and which will be denoted as the non-genomic functions of or effects of estrogen. And when they will bind with these cell membrane receptors, they will act through via the protein kinase or MAP kinase pathways. So now it is the time to discuss the hormone progesterone, next hormone under all these 6 headings, that is their synthesis or structure and synthesis, their secretion and metabolism, functions and mechanism of action. So progesterone, it is a C21 steroid, and it is secreted from the corpus luteum, we know that, and it is also secreted from the placenta. And it is an important intermediate, so we can say that it is an important intermediate of the steroid biosynthesis. We know that when the cholesterol, they will enter into a cell, so this cholesterol will enter into the mitochondria, where this will be converted to pregnenolone. Then again it will come out and it will form what? that is the progesterone. From the progesterone there may be generation of cortisol, corticosterone, androgen, so many things, but the initial, this is a very important intermediate of the steroid hormone synthesis. So small amount apparently enter the circulation from testis and the adrenal cortex. Now we know that this is a function of progesterone. It will help the placenta and it will also help the newly developed fetus to be maintained and to grow. Now progesterone synthesis, we know that this will follow again that previous two-cell steroidogenesis theory, that is from the theca cell the cholesterol will be converted to the androstenedione, which will be transferred to the granulosa cell, which contains the enzyme that is aromatase, which will ultimately forms the estradiol. We know this concept, and in between them we know this is the hormone which is produced. This is a progesterone. So this progesterone will also be released in the corpus luteum cell. In the corpus luteal cell, so this progesterone will be released into the blood. So this is, you can see the LH, which is causing the secretion of the androgen, and everything is happening in the previous way. And the intermediate of this process, here it is a progesterone, and this progesterone will be released from the circulation. Now if we look into the level of progesterone at different phases of menstrual cycle, and we know that when the corpus luteum is formed, at this phase the progesterone level, its concentration, it rises at the post-ovulatory phase, that is at the luteal phase. So LH, when it binds with the LH receptor at the thecal cell, and the progesterone secretion will be increased due to activation of this adenyl cyclase and everything. So what is the level of this progesterone? Progesterone has a very short half-life, and you can see in the early follicular phase the level is 1. In the pre-ovulatory phase this value is 4 milligram in 24 hours per day, and in the mid-luteal phase the value is 25. So there is huge increase in the mid-luteal phase. Now how much is this value? It is around 80 nanogram per ml. So just like estrogen, we'll see that total progesterone, it will remain in two forms. One is a free form, only 2%, and another will be the bound progesterone, that is 98%. And the bound progesterone, it is in the bound with the albumin in 80%, and the steroid binding globulin in 80%. So regarding the metabolism of progesterone, we find that progesterone has a very short half-life, and it is converted in the liver. You will see that it will be converted in the liver to the pregnenolone, which is conjugated to glucuronic acid and excreted in the urine. So this is a mechanism. It will be converted to pregnenolone, and which is conjugated with the glucose. So this is your pregnenolone, and it will be conjugated with the glucuronic acid and excreted in the urine. Come to the functions of progesterone. Progesterone has got main 3 target organs, as we all know. It will act on the female reproductive system, it will act on the breast, and it will act on the brain. So it decreases the uterine motility. So we know that during the time of pregnancy, when the blastocyst has been implanted on the uterus, so it is now the time to keep this blastocyst in a peaceful condition. So all type of uterine excitability has to be reduced. So uterine motility will be decreased with the help of progesterone, and it will also reduce the excitability and it will by reducing the membrane potential, it actually reduces the excitability, and it also reduces the number of estrogen receptor. So it also helps in the development of the secretory endometrium. We know that all this spiraling of arteries and everything will be there. Thickens the cervical mucus, development of breast growth, that is the alveolar development, increase in the body temperature, that is the reason. So after ovulation, there will be basal body temperature will be raised, increased appetite, depressed T cell function, anti-insulin effect. These are the functions of progesterone in the body. Now the mechanism of action of progesterone, like other steroid hormone, again it will bind with the nuclear receptor. Now the speciality of nuclear receptor is that the progesterone receptor is bound with the heat shock protein or HSP. It is bound with the heat shock protein. Okay. When actually this progesterone receptor, it contains heat shock protein. When the progesterone binds with its receptor, this heat shock protein will be removed and the DNA binding sites will be exposed. So it will bind with the DNA, and there will be transcription of mRNA, and there will be protein synthesis, the same like the nuclear receptor superfamily. Now one important finding is that there are certain chemical agents, for example this one, this mifepristone, which are also known as the SPRM, that is selective progesterone receptor modulators, SPRMs. Okay, what actually they do? So they binds with the progesterone receptor, but it does not release the heat shock protein, and it blocks the binding of the progesterone. So mifepristone is a kind of antagonist of progesterone. So it will bind in place of progesterone, it will bind with it. So it will compete with the progesterone to bind with this receptor. But we know that progesterone, when bind with this receptor, the heat shock protein will be released, and it will be able to bind with the DNA. But mifepristone, when it binds with the receptor, it will not release the heat shock protein, and the action of progesterone will not happen. So it is preventing the progesterone to bind with this receptor. It binds to the receptor by itself, but it is inhibiting the functions of progesterone also. So it is an antagonist of progesterone activity. It is mifepristone is also known as RU486. What is the clinical importance of that? We know that progesterone is very, very important for maintenance of pregnancy. So any progesterone antagonist will prevent this maintenance. Now the maintenance of early pregnancy, it depends on the stimulatory effect of progesterone on endometrial growth or its inhibition of uterine contractility. So it is seen that mifepristone and a combination of mifepristone and the misoprostol is the prostaglandin. Prostaglandin is very much excitable to the uterine musculature. So this combination can be used for elective abortion. This is a clinical use of this drug. So now another hormone that comes out of the ovary, that is the relaxin. The corpus luteum secretes a second peptide hormone called relaxin. As the name implies, in human it relaxes the myometrium and plays an important role in the parturition. So during the time of pregnancy, it will keep the myometrium relaxed. But during the time of parturition, it will cause softening of the uterine cervix, which will promote the movement of fetus in the forward direction. So it will help in the process of labor. Relaxin is encoded in two genes, H1 and H2 on the chromosome 9. Although both relaxin genes are expressed in prostate, only the H2 gene is expressed in the ovary. A physiological role of relaxin in the non-pregnant woman has not been established. Thank you.